Okay, good morning, students. Is my slide visible to you?
Yes, sir.
Okay, thank you.
Yes, sir.
Now, listen, in the last lecture, I had started with this unit, animal tissue culture, I had given you introduction regarding animal tissue culture. So, I talked about what is meaning of animal tissue culture, what it includes. So, I told you that it includes culturing of cells, tissues, as well as organs. And what is meaning of culture medium? What is alternative name for culture medium? What is present in culture medium? That was discussed, and who had done first tissue culture, that was told to you. Then how organ culture is done, general idea regarding that was given to you. Okay, I talked about scaffold. Okay, then introduction about aseptic technique was given. What is meaning of aseptic technique? What is sterilization? Okay, what is aim of aseptic technique? Aim means objectives. Okay, then which measures should be followed to avoid contamination? Those were discussed. After that, I had discussed some terms related to sterilization. So these were those terms which you can see on screen. And now we come to the next part. Okay. So next topic is sterilization in this unit. Okay. So can anyone explain me what is meaning of aseptic technique? Sterilization is one method or one type of aseptic technique. So, what is aseptic technique? In the last lecture, the explanation was given regarding meaning of aseptic technique. All the methods or techniques which are used for avoiding contamination are known as aseptic technique. Okay, so all the methods or procedures which are followed for avoiding contamination in tissue culture are called aseptic technique. Sterilization is one method or one type of aseptic technique. So this prevents contamination. That's why it is one type of aseptic technique. Remember this. So what is sterilization? We'll see now. Sterilization refers to, sterilization refers to any process that eliminates, removes, kills or deactivates all forms of life and other biological agents present in specific region, surface, or culture medium. So, in short, I can say that it is a method which eliminates or removes or kills all forms of life from a particular area. And that particular area can be a culture medium, it can be any surface or any region, it can be your working table in the lab. Okay. So, I suppose this concept of sterilization is clear to you. And what is meaning of biological agents? In bracket, I have mentioned it includes bacteria, fungi, spore forms. Listen, viruses are not living organisms, but they are also included here. Okay. So when you are doing sterilization, you make any surface free of viruses also. For example, nowadays we use hand sanitizers. Okay. So in hand sanitizers, mostly 70% alcohol is present. And that 70% alcohol can kill all these life forms, including viruses. Listen, virus is not a life form, it is not a living organism. But due to 70% alcohol, this virus gets destroyed because most of the viruses are made up of lipids and proteins. And when you treat those molecules with alcohol, they get destroyed. Okay, and that's why viruses are also killed due to sanitizer. So remember this. So this is meaning of sterilization, making any place or anything free from life forms is sterilization. Simplest definition. So you can note down. It is a method, sterilization is a method of making anything or any place free from free from life forms. Free from life forms. So if life forms are present, they are eliminated or killed in this method. So I'm proceeding further. Now see how to carry out sterilization. See, sterilization can be achieved through various means such as heat, chemicals, irradiation, high pressure, and filtration. Okay, listen. So these are different means or methods for carrying out sterilization. Okay, so if you want to carry out sterilization, these are the different options available to you. You can use heat, you can use chemicals. अभी मैंने बताया एक अल्कोहल का example. You can go for irradiation. You can use different radiations like UV light. Okay, UV light can kill microbes. It can kill any life form or high pressure, or you can use particular filter if you want to sterilize a liquid. Okay, so you can go for using filter. Now, what is importance of sterility in cell culture? So if you are sterilizing a particular place or particular culture medium, then what is use of it? Why to sterilize? Okay, the answer is very clear. See, creating a sterile field is difficult, but it is important in tissue culture. Why? Because if you're working in sterile area or sterile field, then what will happen? No contamination will occur. Okay, or the chances of contamination would be very less. Chances would be very less. So if you're working with a particular cell type, for example, if you are working with pancreas cells, okay, and you don't want any type of contamination, then you should have a sterile working area. Sterile working area means free from any life form. Okay, so if you're working in sterile working area, then chances would be very less that any other living life form enters in the cell culture of pancreas cells. If you're working with diplococcus bacteria and you don't want any other bacteria or fungus to enter in that culture, then you should have sterile working area or sterile facility. Okay, remember this. Now, when do we require sterile condition? Okay, so sterile condition is required during transfer of cultures. Suppose you are transferring particular type of cells from one culture vessel यानी जिसमें तुम ग्रो कर रहे हो एक फ्लास्क से दूसरे फ्लास्क में ट्रांसफर करना है समझो. So if you want to transfer a particular type of cell from one culture vessel to another culture vessel, then you require sterile condition. You require aseptic technique. Okay. Next is inoculating media. Suppose you want to grow a particular type of cell in your lab. You have brought some samples, okay, and you want to grow that sample in nutrient medium. So when you are introducing sample or a particular type of cell inside nutrient medium, then that process is known as inoculation. That process is known as inoculation. So when you are inoculating media, then also aseptic technique or sterilized condition is required. Sterile environment is required. So these are the situations where we need aseptic technique or sterile condition. Now third, isolation of pure culture. Okay. So what is this? Suppose you want to separate out a particular type of bacteria from a group of bacteria which are growing in nutrient medium. Then for that also you need sterile condition. Then next, for performing microbial tests. So for performing different types of microbial tests, this is required. So you want to grow a particular type of microorganism. You don't want other microorganisms to disturb that culture. Then you need aseptic technique or sterile condition. आई होप आई एम क्लियर. किसी को डाउट है ये 4 पॉइंट्स में से किसी में भी तो बोलो. इनोक्यूलेशन का मीनिंग लिख के रखो. इनोक्यूलेशन, इंट्रोडक्शन. इंट्रोडक्शन फॉर कर्ड मेकिंग ऑल्सो वी इनोक्यूलेट बैक्टीरिया. ओके, इन द मिल्क. तो पहले का जो कर्ड रहेगा उसमें से थोड़ा सा कर्ड लेके तुम फ्रेश मिल्क में ऐड करते हो. दैट इज ऑल्सो नोन एज इनोक्यूलेशन. इनोक्यूलेशन मीन्स इंट्रोडक्शन. Okay, next point. Proper aseptic technique prevents contamination of cultures from foreign bacteria inherent in the environment. So, in the air which we breathe, in that air also, many different types of bacteria are present. The workplace, where do we work? There also many different types of microbes are present. So, to prevent contamination of cultures यानी क्या? The cells which are grown in the lab, in the culture vessels are called cultures. The cells which you grow in the culture medium or inside the culture vessels, those cells are called cultures. ध्यान में रखो, group of cells को बोलते cultures in tissue culture. Okay, so for preventing contamination of cultures from foreign bacteria inherent in the environment, present in the environment, we require proper aseptic technique. So note down meaning of this term. Last time भी मैंने बोला था, note down करके रखो, नहीं तो बाद में भूल जाओगे. What is meaning of cultures? So, right, in tissue culture, this term is used for a group of cells. This term is used for a group of cells. Okay. Other important points related to aseptic technique, why it is required. Aseptic technique is of utmost importance to maintain pure stock cultures while transferring cultures to a new media. Okay, so this has been discussed already. Pure stock culture, that means cells of single type. Pure stock cultures means cells of single type. Cells of same type बोला तो भी चलेगा. So you are having a particular type of cell inside one culture vessel or inside one flask. Okay, and you want to transfer that particular type of cell into another flask for multiplication. ओके, तो दूसरा जो फ्लास्क है तुम्हारा, उसमें तुमने न्यूट्रिएंट मीडियम लिया हुआ है, उसमें एक भी सेल नहीं है. तुम ओल्ड वाला जो फ्लास्क है उसमें से देखो, ओल्ड जो कल्चर है उसमें से थोड़े सेल्स लोगे और इनोक्यूलेट करना चाहते हो न्यू वाले फ्लास्क में जिसमें फ्रेश कल्चर मीडियम है. और वो इनोक्यूलेट करने से क्या होगा, दो सेल्स विल मल्टीप्लाई इनसाइड फ्रेश न्यूट्रिएंट मीडियम एंड दे विल इंक्रीज इन नंबर. तुमको ऐसा करना है, और लेकिन उसमें कंटामिनेशन नहीं चाहिए. तुम यू वांट प्योर स्टॉक कल्चर, सेम टाइप के सेल चाहिए, बाहर का कोई फॉरेन सेल नहीं चाहिए. सो फॉर दैट यू रिक्वायर aseptic technique. If you are following proper aseptic technique, then you will be able to maintain pure stock culture. So note down meaning of this: pure stock culture, cells of same type or single type लिखा तो भी चलता है. So for this, aseptic technique is required. Next point. Aseptic technique is also essential for isolation of single species of microorganism from a mixed culture to obtain a pure culture. तो ये मैंने ऑलरेडी बताया है, ग्रुप ऑफ बैक्टीरिया, उसमें से एक पर्टिकुलर टाइप ऑफ बैक्टीरिया तुमको सेपरेट करना है, तो उसके लिए तुमको एसेप्टिक टेक्निक लगेगा, क्योंकि तुमने सेपरेट तो कर लिया ग्रुप में से, और उसको जहाँ पे ट्रांसफर किया, अगर एसेप्टिक कंडीशन मेंटेन नहीं किया है, तो बाहर के जो बैक्टीरिया है, एयर में है या फंगस है, वो सब इंट्रोड्यूस हो जाएंगे. सो for this type of separation also, we need aseptic technique for isolating a single species of microbe from a group of microbes. Next, next use of aseptic technique. Proper aseptic technique prevents microbes used in the lab from accidentally being released into the environment. This is also very important point. If you are using proper aseptic technique, then this should not happen, that the microbes on which you are working in the lab, they are getting released in the environment. This should not happen. Otherwise, many pandemics or epidemics may arise in the particular area. Epidemic यानी एक particular area में restricted है. Pandemic यानी पूरे world में spread हो गया, तो बोलते हैं pandemic. If you're working on a very deadly microbe, जैसे कि देखो, एक अभी एग्जांपल सामने ही है, कोरोना वायरस. ओके, so it is assumed by many scientists that some scientists of China were working on this virus, and accidentally this virus got released in the environment. And from there it has spread to entire world, and it has resulted in COVID-19 pandemic. So if proper aseptic technique is followed, then this does not happen. This doesn't happen. Remember this. If you are not following proper aseptic technique, then this may happen, and many pandemics and epidemics would occur. Okay. See, this is very important when pathogens are being handled. Pathogens यानी disease causing agents. Disease cause करने वाले agents तुम handle कर रहे हो, तो बहुत important है. Remember this. There is one bacteria by name botulinum. Okay, botulinum, and that bacteria produces a toxin by name botulinum toxin. Only 5 ml culture. Listen, very important point. Only 5 ml culture of that bacteria can kill entire humanity. Remember this. Only 5 ml culture of that bacteria can kill entire humanity. सब humans का death हो जाएगा अगर वो 5 ml से release हो जाए world में तो. And scientists work on many such bacteria and viruses which are very deadly. So for handling those deadly pathogens, aseptic technique is must. Okay. Now I'm going to talk about types of sterilization. Okay. So you know meaning of sterilization. It is one type of aseptic technique, and why aseptic technique or sterilization is required, that has been discussed just now. Now I'm going to talk about types of sterilization. Okay. So sterilization is of three main types: physical, chemical, and filtration. So in the introduction part, I had talked about this. So these are three main types of sterilization: physical sterilization, chemical, and filtration. Physical sterilization, see the name. Physical sterilization is done by using physical agents like heat, radiation. Heat and radiation, दोनों physical agents. Okay, chemical sterilization is done by using different chemicals. Can anyone give me any example of chemical which can be used for sterilization? देखते हैं, सब लोग जाग रहे कि सो गए. Give me example of any one chemical which can be used
Al-
for sterilization. Yes,
alcohol.
very good. Alcohol. Okay. Then filtration. For filtration, different types of filters are available. So I will discuss in detail about these in the next slides. So first, physical sterilization. So in the beginning itself, I told you that physical sterilization is done with the help of physical agents. Okay. And this includes physical agents include heat and radiation. So one by one, I will discuss those. See, this class includes heat sterilization and radiation sterilization. तो फिजिकल स्टेरिलाइजेशन में ये दो टाइप है, हीट एंड रेडिएशन. और किसके बेसिस पे ये ऐसे नेम्स है, फिजिकल केमिकल फिल्ट्रेशन, बेस्ड ऑन द एजेंट्स विच आर यूज्ड फॉर स्टेरिलाइजेशन. यहाँ पे फिजिकल एजेंट्स यूज होते हैं. चलो जरा ये लिख लो हेडिंग, टाइप्स ऑफ स्टेरिलाइजेशन में, तो आगे जाके कंफ्यूज हो गए तुम लोग. राइट दिस हेडिंग, टाइप्स ऑफ स्टेरिलाइजेशन, एंड अंडर दैट राइट based on based on the agents which are used for sterilization, based on the which agents which are used for sterilization, comma, the different types of sterilization are as follows. The different types of sterilization are as follows. एंड अभी नेक्स्ट लाइन में लिखो फिजिकल स्टेरिलाइजेशन, केमिकल स्टेरिलाइजेशन, और सबके सामने छोटा ऐसा लिख सकते हो शॉर्ट डिस्क्रिप्शन. फिजिकल स्टेरिलाइजेशन के सामने लिखो फिजिकल एजेंट्स आर यूज्ड. केमिकल स्टेरिलाइजेशन, केमिकल एजेंट्स आर यूज्ड. फिल्ट्रेशन के सामने लिखो फिल्टर्स आर यूज्ड. बी क्विक. फुल लिखो, सिर्फ ऐसा फिजिकल केमिकल मत लिखो, फिजिकल स्टेरिलाइजेशन. तो फिजिकल के सामने लिखो फिजिकल स्टेरिलाइजेशन के सामने फिजिकल एजेंट्स आर यूज्ड. केमिकल स्टेरिलाइजेशन के सामने chemical agents are used, and filtration के सामने different types of filters are used. Okay, now next slide. Now see, under physical sterilization two different types are there: heat sterilization and radiation sterilization. So what is heat sterilization? Name से ही इसका meaning समझ में आता है. देखो, heat sterilization procedure is the one in which destruction of microorganisms mainly occurs due to the high temperatures employed. So heat sterilization is that sterilization in which microorganisms are destroyed with the help of heat. Simple definition. Microorganisms are destroyed with the help of heat. या ऐसा तुमने बोला, it is a type of sterilization in which microorganisms are destroyed with the help of heat, तो भी चलेगा. So remember this. So how it is done? See, heat can be of two types. Heat which is used for sterilization can be of two types: moist heat and ड्राई हीट. अब ये मॉइस्ट हीट एंड ड्राई हीट में क्या डिफरेंस है? लिसन, अगर वाटर वेपर्स के हेल्प से तुम कुछ कोई माइक्रो ऑर्गेनिज्म को डिस्ट्रॉय कर रहे हो, वाटर वेपर्स एट वेरी हाई टेम्परेचर, ओके, तो उसको बोलेंगे मॉइस्ट हीट स्टेरिलाइजेशन. फॉर एग्जांपल, प्रेशर कुकर सबने घर में देखा रहेगा. जब उसमें से देखो एकदम हाई प्रेशर बिल्टअप होने की वजह से विसल जब ब्लो होता है उसका, व्हेन whistling is done by pressure cooker, at that time water vapors are released by the pressure cooker from the whistle area, and those water vapors are released under very high pressure. Okay, so and those water vapors are very hot If you come in contact with those water vapors, your skin may get burned. Okay, so that is one example of moist heat. So if your skin cells can die due to contact with that moist heat, then microbes can also get killed with the help of that water vapor. Okay, so similar to pressure cooker, in our labs we use one instrument which is known as autoclave. The working principle of this instrument is similar to the working principle of pressure cooker. Here also water is added in this instrument. Okay, and water is heated and the lid is closed. Listen, due to closed lid, what happens? Pressure builds up. Gradually, pressure increases inside this instrument like pressure cooker. जैसे प्रेशर कुकर का भी लिड बंद करने के बाद प्रेशर इंक्रीज होने लगता है. And when pressure increases too much, then here also one whistle is available. Through that whistle, we come to know that a particular level of pressure has been achieved. And on this instrument, pressure gauge, प्रेशर मेजर करने के लिए एक छोटा सा इंस्ट्रूमेंट रहता है, वो उसको बोलते हैं प्रेशर गॉज. प्रेशर गॉज इज ऑल्सो प्रेजेंट, एंड by looking at the pressure gauge, you can make out how much pressure has built up inside. Okay, and you can introduce your culture medium. Okay, the glasswares and other instruments which are used in lab for tissue culture inside this instrument autoclave, and all of them will get sterilized due to moist heat which gets created inside the autoclave. In college, in third lab, you must have seen this. एक बड़ा सा cylindrical container जैसा है और नियर द विंडो रखा रहता है, ये स्टील का रहता है, शाइनी स्टील. ओके, so which condition is maintained inside the autoclave? Okay, so which condition is maintained inside the autoclave? Listen, this particular pressure is maintained 15 lb. Lb means pounds per square inch. PSI per square inch. So this much pressure is needed. 121 degree Celsius is required, and this particular pressure and this particular temperature is maintained minimum for 15 minutes. If you are keeping your instruments and culture medium inside this autoclave at this particular pressure and at this particular temperature, minimum for 15 minutes, then only proper sterilization would be done. Remember this. Wait, I will show you photograph. I forgot to paste photograph here. मुझे लग रहा है बहुत लोग को याद भी नहीं होगा लैब 3 में कैसा वो दिखता है. Okay, now you can see on screen. This is autoclave, and here pressure gauge is present. Okay, and this pressure regulating device is there, जो अपना pressure cooker में whistle के ऐसा रहता है. This is lid. Okay, so we are having big sized autoclave in our college. ये small sized autoclave है, and this is used for sterilization. And which type of sterilization? Moist heat sterilization. Why moist heat? Because with the help of water vapors in high pressure, sterilization is done by this instrument. So remember this. So this is about autoclave. Can anyone tell me which pressure and which temperature is needed for proper sterilization by using this instrument? अभी मैंने बताया था. Values याद रखना है. How much pressure is needed?
15
Yes.
IBL.
Yes, 15 lb per square inch और 15 psi बोला तो भी चलेगा. 15 psi, 15 lb psi, 15 pounds per square inch. Okay, how much temperature is needed?
1 to 1 degree Celsius.
Okay, very good. And for how much time sterilization has?
15 minutes.
Yes. Now one simple question. Listen, you know that the boiling point of water is 100 degrees Celsius. At 100 degrees Celsius, boiling occurs, and water, liquid water, gets converted into water vapors. Then how do we achieve 121 degree Celsius inside the autoclave? तो 121 कहाँ से अचीव कर लेंगे हम लोग, बताओ? How it is possible to get 121 degree Celsius temperature by using water inside the autoclave? वो water vapor का ही temperature इतना increase हो जाता है. How it is possible? Listen, it is possible due to high pressure. In your physics lectures of lower standards, you must have learned that if pressure increases, then the boiling temperature also increases. Okay, remember this. अगर प्रेशर इंक्रीज किया तो बॉइलिंग टेम्परेचर भी कोई भी सब्सटेंस का इंक्रीज हो जाता है. रिमेंबर दिस, एंड दिस इज द प्रेशर गॉज. यहाँ पे हम लोग देख सकते हैं कि कितना प्रेशर अंदर हुआ है, एंड इस पे टेम्परेचर इंडिकेटर भी रहता है, वो टेम्परेचर भी बताता है. So this was about autoclave. Next, dry heat sterilization. तो थर्ड लैब में देखो एकदम कॉर्नर में रखे हुए हैं ओवन, एंड इवन अपने घर में भी ओवन्स हम लोग यूज करते हैं. सो द ovens which are used in homes, those can also be used for sterilization. Remember this. Now see, dry heat is the simplest and most economical method of sterilization. This method requires higher temperatures and longer exposure times to achieve same microbial killing efficiency as compared with that of steam sterilization. Steam sterilization and moist heat sterilization. Okay, so here it is simple, but here more time and longer exposure is required for killing of microbes. Remember this fact. So, which condition has to be maintained inside oven, hot air oven, for killing of microbes? A minimum of 180 degrees Celsius for minimum 30 minutes. 30 minutes के लिए minimum रखना पड़ेगा. इधर देखो क्या लिखा है, not less than 30 minutes, यानी minimum 30 minutes. या अगर 170 मेंटेन कर रहे हो तो मिनिमम 1 hour रखना पड़ेगा, जो भी इंस्ट्रूमेंट्स को क्लीन करने के लिए रखा है तुमने इनसाइड द ओवन. सो यू कैन रिमेंबर एनी वन वैल्यू. यहाँ पे दिया है 160 के लिए 2 hours रखना पड़ेगा. सो यू कैन सी टू मच टाइम is needed and high temperature is needed for sterilization by using oven. So dry heat sterilization is economical, okay, but it takes more time and it requires more temperature for killing of microbes. Remember these two facts. Now next, radiation sterilization. So here different radiations like UV ray, gamma ray. Okay, so UV rays and gamma rays are used. See, this process is accomplished by exposure to UV light or high energy ionizing radiation such as gamma rays. ओके, एंड एक्सेलरेटेड इलेक्ट्रॉन्स, ये भी यूज होता है फॉर स्टेरिलाइजेशन. ओके, नाउ how these radiations work, that has been mentioned on this slide. Now see, UV rays क्या ऐसा करता है कि microbes kill हो जाते हैं. See, UV rays can damage DNA of microbes by forming dimers of nucleotides. Thymine dimers तुमने last semester में learn किया है. Okay, तो थाइमिन डाइमर्स फॉर्म हो जाएंगे और डीएनए डैमेज हो जाएगा माइक्रोब का, तो ऑटोमेटिकली उसका डिस्ट्रक्शन हो जाएगा, वो किल हो जाएगा. क्योंकि अगर रिपेयर नहीं कर पाया तो उसका नेक्स्ट स्टेप है डेथ. और तुम कंटिन्यूस एक्सपोजर में रखे हो तो उसके प्रोटीन्स वगैरह भी डैमेज हो जाएंगे ड्यू टू यूवी रेज, एंड irreparable डैमेज हो जाएगा, irreparable, which cannot be repaired, and then microbe will die. Okay, now see, when properly operated, the UV radiation technique can reduce the level of airborne bacterial contamination in room by 90% within 30 minutes. यानी अगर तुमने यूवी लैंप ऑन किया, यूवी लैंप देखो, यूवी रेस के लिए यूवी लैंप मिलता है, बल्ब जैसा ही रहता है, वो फिट किया अपने लैब में, तुमने 30 मिनट्स तक ऑन करके रख दिया, लैब को क्लोज करना पड़ेगा. अगर तुम उसके एक्सपोजर में आओगे यूवी लाइट के तो तुम पे भी इफेक्ट आएगा. रिमेंबर दिस. सो फॉर सेफ्टी, रूम इज क्लोज्ड. ओके, अगर कोई दूसरा मेथड अवेलेबल नहीं, ऑप्शन अवेलेबल नहीं है, तुमको यूवी रेडिएशन के अंडर ही वर्क करना है, तो बहुत प्रिकॉशंस लेना पड़ता है. पूरा फुल्ली कवर्ड ऐसा हैंड्स को कवर करना पड़ेगा, और सिर्फ हैंड ही लेके आते हैं, ग्लव्स वगैरह पहनते हैं. ओके, सो कीप इफ यू आर switching on UV lamp for 30 minutes, then around 90% microbes will get killed from your lab. Okay, airborne bacteria. तो अराउंड 90% बैक्टीरिया किल हो जाएंगे. नेक्स्ट, pharmaceutical products are more resistant to degradation by ionizing radiation if they are in powdered form than in liquid form. ओके, सो फार्मा प्रोडक्ट जो भी रहते हैं, यानी ड्रग्स, ये सब वैक्सीन्स, ये सब फार्मास्यूटिकल प्रोडक्ट्स है. ओके, सो इफ दे आर इन लिक्विड फॉर्म, see the sentence, pharmaceutical products are more resistant to degradation by ionizing radiation if they are in powdered form. So if powdered form of pharma products you are using, then they will not get destroyed easily. But if they are in liquid form, then they will get destroyed easily by radiation. Okay. Next, thermolabile drugs such as penicillin. Thermolabile means temperature sensitive. Thermolabile drugs such as penicillin, streptomycin, thymine, this is vitamin, riboflavin have been effectively sterilized by ionizing radiation. So these can be sterilized by using radiation because these are thermolabile, temperature sensitive है. तो अगर हाई टेम्परेचर से इनको स्टेरिलाइज करने जाओगे तो डिस्ट्रॉय हो जाएंगे. तो उनके लिए इजी मेथड है रेडिएशन. रेडिएशन से हाई टेम्परेचर क्रिएट नहीं होता है. So you can sterilize these by using radiation. देखो, पेनिसिलिन एंटीबायोटिक का नाम है, स्ट्रेप्टोमाइसिन एंटीबायोटिक का नाम है, थाइमीन एंड राइबोफ्लेविन विटामिन बी कॉम्प्लेक्स के मेंबर्स हैं. ओके, तो यहाँ पे मैं क्या बता रहा हूँ, ध्यान देना यहाँ पे कि किसको स्टेरिलाइज नहीं करना है. ओके, लिक्विड फॉर्म में अगर फार्मा प्रोडक्ट है तो स्टेरिलाइज नहीं करना है. तो किसको स्टेरिलाइज कर सकते हैं बाय यूजिंग यूवी रेज, वो यहाँ पे मेंशन है ये वाले पॉइंट में. नेक्स्ट, इन हॉस्पिटल्स UV radiation is used to control the spread of infection during or after surgical procedures. So operations के पहले एंड operations के बाद जो भी सर्जिकल इंस्ट्रूमेंट्स है, वो लोग को यूवी रेडिएशन से क्लीन कर सकते हैं. देखो, तुमने वॉश कर दिया, उसके बाद भी यूवी रेडिएशन के एक्सपोजर में रखते हैं. रिमेंबर दिस. नेक्स्ट इज केमिकल स्टेरिलाइजेशन. तो फिजिकल में देखो, तुमने हीट स्टेरिलाइजेशन पढ़ा, उसमें दो लर्न किया तुमने, moist heat sterilization and dry heat sterilization. Can anyone tell me dry heat sterilization can be done by using which instrument?
Oven like
Yes, oven. Okay. So oven can be used. And for radiation sterilization, which rays are mostly used? Which agents are
Gamma.
used? Yes, gamma rays, then UV rays. Now next question, how UV rays kill the microbes? How do they act on the microbes? अभी मैंने बताया था एक स्लाइड पे. How do they act? How UV radiation acts? It acts on DNA mainly, and it forms what?
Dimers of nucleotide.
Yes, and mostly dimers of which nucleotide are formed? वो भी मैंने बताया था, thymine.
Thymine.
Yes. So when DNA gets damaged excessively due to formation of dimers, okay, and when proteins are also damaged due to UV radiation, then automatically microbes get killed. Okay. So after physical sterilization, next is chemical sterilization. See, this process involves exposure of materials to chemical agents such as ethylene oxide, name of chemical, ethylene oxide, formaldehyde, lutheraldehyde, and propylene oxide. These are names of chemicals which can be used for chemical sterilization. Now next, gaseous sterilization. Listen, some chemicals are used in gaseous form also. See, gaseous sterilization is accomplished by exposure to a gas that kills microorganisms. The most commonly used gas for sterilization is ethylene oxide. So remember, this is most commonly used gas in labs for sterilization. So मैंने bold में रखा है, ध्यान देना. Ethylene oxide acts by alkylation. Alkylation is one form of DNA damage. Last semester में हमने learn किया है. अल्काइल एजेंट यानी मिथाइल इथाइल ग्रुप ये सब ऐड हो जाता है न्यूक्लियोटाइड्स में, एंड वो नॉर्मल न्यूक्लियोटाइड अब नॉर्मल न्यूक्लियोटाइड में कन्वर्ट हो जाता है. ओके, नाउ नेक्स्ट, फेनॉल्स. Phenols and phenolic compounds produce variety of effects on the microorganisms. देखो लैब में तुमने phenol नाम का एक केमिकल यूज किया है. ओके, so that can destroy many microbes. Some phenol based soaps and hand washes are also available in the market. For example, लाइफ बॉय, सबने सुना रहेगा लाइफ बॉय, वो फेनॉल बेस्ड हैंड वॉश एंड फेनॉल बेस्ड सोप है. लाइफ बॉय ध्यान में रखो. ओके, बेस्ड ऑन देयर कॉन्सेंट्रेशन, बेस्ड ऑन देयर कॉन्सेंट्रेशन कितना परसेंट वाला फेनॉल यूज कर रहे हो, 10%, 2%, 3%. ओके, based on their concentration, they exert various effects like disruption of cells, disruption यानी breakdown of cells, precipitation of cell protein, protein को destroy किया तो उसको बोलते हैं precipitation of proteins. जैसे कि देखो एग ऑमलेट बनाते टाइम जो भी प्रोटीन तुम एग के अंदर देखते हो वो लिक्विड फॉर्म में रहता है ट्रांसपेरेंट जैसा, और हॉट पैन के ऊपर जाते ही वो पूरा सॉलिडिफाई हो जाता है, उसका वो ट्रांसपेरेंट नेचर लूज हो जाता है. दैट इज ऑल्सो वन एग्जाम्पल ऑफ प्रेसिपिटेशन, डैमेज, डिस्ट्रक्शन ऑफ प्रोटीन्स. तो फेनॉल्स क्या करते हैं, दे कैन डिसरप्ट सेल्स, they can cause precipitation of cell proteins, they can cause inactivation of enzymes and leakage of amino acids from the cells. And by doing all this, they help in destruction of microbes. So how phenols act, that has been discussed here. Okay, how ethylene oxide acts, that has been discussed in the first point. ध्यान दो यहाँ पे important points क्या है. Next, other chemical agents include alcohols, halogens. Okay, halogens सबको पता है, कोई मुझे बताएगा halogens का मीनिंग क्या है? केमिस्ट्री बैकग्राउंड वाले स्टूडेंट्स को बेटर वे में पता होगा halogens. कोई एक एग्जांपल दो halogen का.
Chlorine.
वेरी
Br. Bromine.
वेरी गुड. क्लोरीन, ब्रोमीन, आयोडीन, ये सबको हैलोजेंस बोलते हैं. ये एग्जांपल्स हैं. तो अगर हैलोजेंस यूज किया, heavy metals and their compounds, ये भी sterilization में use होते हैं. Okay. Dyes and detergents. Some dyes and detergents also have sterilization property. Then quaternary ammonium compounds and aldehydes. तो इतना सब याद रखने की जरूरत नहीं है. एटलीस्ट तुम 2-3 एग्जाम्पल्स याद रखो. Chemical agents which can be used for sterilization. Okay. Now next method of sterilization is filtration. Filtration sterilization. See, this method is used for sterilizing thermolabile solution. जो temperature sensitive है, उसको अगर तुम heat treatment दोगे तो destroy हो जाएगा, उसका property ही destroy हो जाएगा, property change हो सकता है. So in such cases, we go for either radiation sterilization or filtration sterilization. I'm repeating, for sterilization of temperature sensitive material, we either go for filtration sterilization or for radiation sterilization. See, the solutions are passed through the sterile bacteria-proof filter unit and subsequently transferred aseptically into the sterile containers, which are then sealed. तो देखो अभी किसी सॉल्यूशन में तुमको डाउट, अभी समझो तुमने न्यूट्रिएंट मीडियम प्रिपेयर किया एंड तुमको ऐसा डाउट आ रहा है कि इसमें बैक्टीरिया कोई ग्रो हो गया है एंड यू डोंट वांट दैट बैक्टीरिया, तो क्या कर सकते हो, यू कैन पास दैट सॉल्यूशन थ्रू ए फिल्टर. ओके, एक फिल्टर के थ्रू पास किया तो बैक्टीरिया, देखो अगर बैक्टीरियोलॉजिकल फिल्टर रहेगा तो बैक्टीरिया को ट्रैप कर लेगा. ओके, एंड खाली तुम्हारा न्यूट्रिएंट मीडियम उसमें से पास होके बाहर आ जाएगा एंड यू कैन कलेक्ट दैट न्यूट्रिएंट मीडियम इन another container and that nutrient medium can be used. To sterilize a product, it is often necessary to combine several types of filtration to achieve removal of microorganisms. तो देखो सेफ्टी के लिए सिर्फ एक बार फिल्ट्रेशन नहीं करते, डिफरेंट टाइप्स ऑफ फिल्टर रखते हैं लैब में. एक बार तुमने समझो बैक्टीरियोलॉजिकल फिल्टर से पास किया, हो सकता है और भी उससे भी फाइन मैश साइज वाला तुम्हारे पास और एक फिल्टर है बैक्टीरियोलॉजिकल. देखो उसमें मैश साइज अलग अलग रहता है. मैश साइज तुमने बॉटनी में प्रैक्टिकल या जूओ में भी देखो, एसवाई में सॉइल एनालिसिस किया था तो अलग अलग साइज के मैशेस तुमने यूज किए थे. 0.1 मिलीमीटर जिसका था साइज, 0.5 मिलीमीटर, ओके, 0.001 मिलीमीटर, ऐसा अलग अलग मैश साइज का तुमने मैशेस यूज किए थे. तो वैसे ही डिफरेंट मैश साइज के फिल्टर्स भी आते हैं कि जो 0.1 माइक्रोमीटर तक के बैक्टीरिया को ट्रैप कर लेगा, ऐसा आएगा. ओके, कोई रहेगा 1 माइक्रोमीटर तक के ऑर्गेनिज्म को ट्रैप कर सकता है. सो डिफरेंट टाइप्स ऑफ फिल्टर्स आर अवेलेबल. आई एम हैविंग पिक्चर ऑफ वन ऑफ देम. दिस इज मिलीपोर फिल्टर. ओके, देखो ये फिल्टर पेपर जैसा ही दिखता है एंड इसका फिल्टरिंग यूनिट ऐसा है. तो यहाँ पे रख देते हैं एक पेपर को एंड जो लिक्विड को फिल्टर करना है वो ऊपर से तुम इंट्रोड्यूस करते हो और ये फिल्टर में देखो यहाँ पे ये, यहाँ पे देखो ग्रेजुअली क्या होगा, ये जो भी तुम्हारा मीडियम है ये वो इसमें से फिल्टर होके नीचे आएगा, इधर एक्यूमुलेट होगा और बाद में ये देखो ये पूरा खाली हो जाएगा एंड ये क्लीन लिक्विड है एंड दिस कैन बी यूज्ड. ओके, सो दिस इज वन एग्जांपल ऑफ फिल्टर. दिस फिल्टर इज नोन एज millipore filter. So many such filters are available for use in tissue culture labs. So I'm done with discussion of sterilization. Okay, can anyone tell me different types of sterilization? Types, 3 टाइप हमने डिस्कस किए. मेन टाइप्स का नाम मुझे बताओ.
Physical sterilization,
Yes,
chemical sterilization,
yes,
filtration sterilization.
yes. If I'm having thermolabile material to be sterilized, I want to sterilize a thermolabile material, then which sterilization would be better out of these 3?
फिजिकल
थर्मोलैबाइल, हाँ फिजिकल में.
filtration.
अच्छा, अगर फिजिकल स्टेरिलाइजेशन ही मुझे यूज करना है तो उसमें से कौन सा मैं यूज करूंगा फॉर थर्मोलेबाइल मटेरियल्स? समझो मेरे पास फिल्टर्स नहीं अवेलेबल है।
Radiation.
Radiation.
वेरी गुड। ओके, आई थिंक सर हैज जॉइंड।
गुड मॉर्निंग सर।
गुड मॉर्निंग सर।
सर इन लोग के पास तो बहुत फिल्टर है।
हाँ।
सब वो स्नैपचैट से लेके कुत्ता बिल्ली गधे
हाँ।
घोड़े सारे फिल्टर है इन लोग के पास।
फिल्टर सप्लाई करके फोटो लेते हैं ये लोग।
हाँ